Welcome to this lecture on research methodology. Today, we're going to explore a foundational topic for any scientist: developing a research plan. Whether you are studying animal behavior in the field or conducting genetic tests in a lab, your research plan is your ultimate guide. Let us dive in and see how we can transform a vague curiosity into a concrete, actionable scientific journey. To begin, imagine you are about to embark on a massive cross-country road trip to a place you have never been before. You would never just jump into your car and start driving blindly without a map, a budget, or a clear destination. In the academic and scientific world, conducting a study without a plan is exactly like driving blindfolded. Developing a research plan acts as your ultimate scientific roadmap. It ensures you know exactly where you are going, what tools you need, and how you will overcome obstacles along the way. Now that we understand what a research plan represents conceptually, let us look at how it actually begins. A research plan is a comprehensive, structured document, and its very first formal step is problem identification. You can't research everything at once. The researcher must narrow down a broad area of interest into a highly specific, testable question. Without this laser focus, your study will wander aimlessly. This specific question becomes the anchor for every single decision you make in the subsequent phases of your scientific inquiry. Once you have your specific question anchored, the next logical step in the plan is defining your experimentation strategy. This is where you decide the physical and methodological context of your study. Will you conduct your research in a highly controlled laboratory environment where you can manage every variable? Or will you observe subjects in a natural field setting, which might be more realistic but harder to control? Specifying this strategy in advance is crucial because it dictates the kind of equipment, permissions, and timelines you will need to successfully complete your work. With the setting chosen, we must now address a critical component of the research plan: the experimental and sample designs. This part of the blueprint dictates exactly who or what will be studied and precisely how they will be selected. You need to define your target population clearly. Are you studying a specific species of fruit fly or university students in a particular age group? By laying out these exact elements in advance, the research plan acts as a binding contract that guides every single phase of data collection, preventing bias and confusion later on. Before we move into the mathematical side of planning, we must emphasize why this meticulous preparation is so vital. In the real world, research is incredibly expensive and time-consuming. It is often funded by grants that require strict accountability. Without a solid plan, a researcher might collect the wrong type of data, making months of work completely useless. Furthermore, a documented plan allows other scientists to review your proposed methods for ethical concerns and logical flaws before any actual work begins. Ultimately, this ensures your final results are valid, reliable, and genuinely useful. This brings us to one of the most critical mathematical steps in developing your research plan: determining your sample size. Finding the right number of participants is a delicate balancing act. If your sample is too small, your results will be heavily influenced by random chance and will not accurately represent the real world. On the other hand, if your sample is too large, you will waste valuable time, money, and resources collecting unnecessary data. To find the perfect balance, researchers rely on specific statistical formulas during the planning phase. To calculate that ideal sample size for large populations, we use Cochrane's formula. Let us break down what each symbol means. The variable n represents the ideal sample size we want to find. Z stands for the z-score, which corresponds to your desired confidence level. The variable p is the estimated proportion of the population that has the attribute you are studying, while q is simply 1 minus p, representing those who do not have it. Finally, e represents the margin of error you are willing to tolerate. If you want higher confidence, your z-score increases, which physically means you will need a larger sample size to be more certain of your results. Now that we have our formula, let us see how to apply it by planning a university survey. Suppose you want to study the proportion of students who study more than 20 hours a week across a massive campus. You want to be highly confident, which gives a standard z-score of 1.96. You are willing to accept a small margin of error of 5%, so E equals 0.05. Since you have no prior idea of the true proportion, you assume maximum variability. This means setting both P and Q to 0.5 for the safest estimate. With our parameters defined, let us calculate the exact number of students to survey. We start by substituting our known values into Cochrane's formula. First, we square the z-score of 1.96 to get approximately 3.8416. Next, we multiply the proportions, 0.5 times 0.5, yielding 0.25. Multiplying these together gives a numerator of 0.9604. We then square our margin of error for the denominator, getting 0.0025. Dividing these gives 384.16. Since we cannot survey a fraction of a person, we round up. Our final answer means we must survey exactly 385 students to achieve our desired confidence. Having successfully planned a survey, let us tackle a much higher stakes scenario: planning a medical clinical trial for a new allergy medication. Because this involves human health, you need a much higher confidence level, corresponding to a z-score of 2.576. You also need a stricter margin of error of just 2%, so E equals 0.02. Previous pilot studies suggest about a third of people respond well to similar drugs, so we set P to 0.3 and Q to 0.7. These stringent parameters will significantly impact our required sample size. Let us apply Cochrane's formula again with these stricter medical trial parameters. We substitute our new values into the equation. Squaring the higher z-score of 2.576 gives us 6.6357. Multiplying our proportions, 0.3 and 0.7, gives 0.21. Multiplying these together yields a numerator of 1.3935. Squaring our tiny margin of error gives a very small denominator of 0.0004. Dividing the numerator by the denominator gives 3,483.75. Rounding up, our final answer dictates that the research plan must account for exactly 3,484 patients to ensure scientific validity. Now that we have mathematically determined our sample size, let us visualize how all these steps come together in the overall research plan flowchart. At the very top, you see problem identification, where the specific question is formed. An arrow leads down to the literature review, ensuring the topic has not already been solved. This flows into experimental and sample design, which is exactly where we apply the sample size calculations we just learned. From there, the plan moves into actual data collection and experimentation, and finally concludes at the bottom with analysis and reporting. This diagram shows how a theoretical idea systematically bridges into a real-world investigation. To wrap up our lecture, let us review the key takeaways. First, a research plan is your scientific roadmap. It transforms a vague curiosity into a structured, actionable journey, saving time and resources. Second, determining the correct sample size is a critical mathematical step that prevents your study from being ruined by random chance or wasted effort. Finally, tools like Cochrane's formula allow us to calculate the exact number of participants needed based on our desired confidence and margin of error. By investing time in this planning phase, you protect the integrity of your scientific inquiry and guarantee impactful results.